Nitration of a benzene is an electrophilic aromatic substitution reaction. Right, so here is an aromatic compound, an electrophile will be substituted on the aromatic compound. So we call it electrophilic aromatic substitution, or EAS reaction. That is chapter 18. We'll see that soon in the lecture. Benzene is a weak nucleophile, right? Because it's a very stable compound, stable aromatic compound. It does not react fast, so we call it a weak nucleophile. The pi bonds in the benzene are the nucleophilic sites, right? And nitric acid is a weak electrophile. So these two cannot react if they are weak, right? So we have to somehow make one of these a better reactive reactant, right? So what we do is we use sulfuric acid to make the nitric acid more reactive, right? A better electrophile. And the product of this reaction would be NO2. Let's see how the sulfuric acid activates the nitric acid. It is the structure of the nitric acid, right? It's a neutral compound. It has positive and negative, but overall charge of nitric acid is neutral. Nitric acid is an acid, which means it can donate the proton readily. It's a strong acid. But when you put a stronger acid next to nitric acid will act as a base, right? It's awkward. Nitric acid is a very strong acid, but sometimes it can act as a base. It can take the proton from the sulfuric acid. What happens when nitric acid takes a proton? You get another positive charge, right, on the oxygen. So this is not a stable structure with two positive charges and a negative charge. What happens is the water will immediately leave as a leaving group and gives the NO2. Water leaves and you get the NO2, or nitronium ion. Nitronium ion, that is a strong electrophile. You see the positive charge on the nitrogen? That is an electrophile, right? You don't have so this is a strong electrophile, and that will push the reaction to go forward. You converted the weak electrophile into a strong electrophile. Now the reaction is okay to go forward, right? Let's look at the mechanism of this reaction. In the first step, benzene takes a proton from benzene attacks the electrophile, the nucleophilic site of the benzene. The pi bond attacks the nitrogen of the nitronium ion, and that gives 3 resonance structure for the intermediate. Here is the NO2 substituted compound. This is no longer a benzene, no longer an aromatic compound. The pi bond is broken, right? So the conjugation is broken here. It's not an aromatic compound. You go from an aromatic is a stable form of the compound, to non-aromatic, right? We'll study about these aromatic and non-aromatic nature in the lecture, but for now we get this non-aromatic intermediate, and there should be a positive charge, right? When one carbon loses the electrons, this carbon gains a positive charge. That positive charge is allylic. You see this allylic nature of the positive charge? Allylic carbocation can resonate. It can move through resonance. It moves to this carbon right here. It can move one more time to this carbon, right, through resonance. It is that structure. And after this intermediate, this proton needs to be removed in order to bring back the aromatic nature. Right? So water will act as a base. The water that you got here will act as a base to remove the proton from that carbon, and the electrons will go neutralize this positive charge by forming this pi bond. So you started from an aromatic benzene, you got the aromaticity back, which means the product is stable. Aromatic compounds are stable because of their cyclic conjugation of pi bonds. Right. So this is the mechanism of nitration of a benzene. The nitro group acts as an electrophile and the benzene acts as a nucleophile. Now, the variation of this is, what if you have a substituent on the benzene? If you start from a substituted benzene, you may have some selectivity, some regioselectivity. Let's look at that in the next slide. Okay, here's the methyl benzoate, right? Methyl benzoate, right? Now let's do the nitration on this substituted benzene. The reaction would be similar. I'm going to move this to here. Okay, this nucleophile attacks the nitronium ion that you got as a result of mixing nitric acid and sulfuric acid. Gives this nitronium ion. In the lab, we're actually going to mix these two acids slowly in a separate flask to get this activated electrophile, a better electrophile, right? Okay, the reaction will proceed by the nucleophilic pi bond attacking the nitrogen. Right? Now, when you do this, you get an intermediate with a positive charge on this carbon. Let's see, H3 here. If you put the NO2 there, the positive charge will be on this carbon, which is not good. Why? This group right here is an electron withdrawing group, which means it tries to withdraw the electron density from the benzene ring through resonance, like that. Oxygen is an electronegative atom. It removes the electron density. So eventually this carbon will be partial positive. Now, if you put a positive charge next to another positive charge, that will be really unstable, right? So we don't want to put the positive charge here. That's the key. That's the key why you don't put nitro group on this carbon. Right? There was a question on the back of the lab manual. Why do you get the nitro group on the meta position but not the ortho position or para position? You don't get nitro groups on these two locations because of the reason that positive charge will end up on a carbon that is next to an electron withdrawing group. Alright, so that's the answer. If you put the nitro group on ortho or para, you will end up with a positive charge next on a carbon next to an electron withdrawing group. Okay, now let's see how this can be fixed by placing the nitro group on a different carbon atom. Now instead of moving the electrons from here, let's move the electrons from here. Right. These electrons are moved to the nitrogen and place the nitro group here, NO2. You can also do that on this carbon, right? We know benzene can be drawn like this or like this. So if this is the benzoate, the C double bond O OCH3, you can move this pi bond or this pi bond to put the nitro group on this carbon or this carbon, right? They're both same. Good. So placing nitro group on this carbon will put the positive charge on this carbon, right? And then this positive charge will resonate to this carbon like this. And to this carbon. So it will the positive charge will never goes to the carbon next to an electron withdrawing group, that is this carbon. So watch the positive charge, it never goes to that carbon. That is because you chose the nitro group to go on to the meta position, right? If the nitro group is on the meta position, the positive charge will never go to the carbon next to electron withdrawing group. Okay, after that, water will take the proton from here, right, H2O, take a proton from there, and in the lab we add extra water in the form of ice to do this step, right? And you'll get the product where nitro group is on the meta position. Okay. The other questions that we need to answer from the back of the lab manual is, so this is the arrow pushing mechanism, that the question number 1 on the post lab. Draw the arrow pushing mechanism. Is that one arrow that goes from the pi bond to the NO2, and then the arrows for the resonance, and finally the arrow for deprotonation, right? That's arrow pushing mechanism. This would also answer question number 2, why the nitro group goes to the meta position, and justify that using the 3 resonance structures. Here are the 3 resonance structures, 1, 2, 3, and the focus is on the positive charge. Where is the positive charge? It never goes to this carbon, right? That carbon. That answers second question. The third question is, why do you mix the nitric acid and sulfuric acid slowly? I think I explained that in the lab demonstration video, that If you mix these two acids, it is an exothermic reaction. Right? It can go bad if you add sulfuric acid suddenly to the nitric acid. It may create a lot of heat and boil off the mixture, and sometimes it can shoot out of the flask. So to contain the exothermic reaction, we need to add slowly. And the last question number 4 on the back of the lab manual: why do you add the nitric acid and sulfuric acid to the methyl benzoate slowly? It is another exothermic reaction. Adding these two, which is this, too fast will create a large exothermic reaction. The other answer could be, if you add too fast, you probably end up getting more than one nitro substitution. The nitro group will be substituted twice, one on here, one on here. Right? So dinitro methyl benzoate could be the other product that is possible. So to avoid that, we add slowly.